Hi, my name's Carly. My groupmate Asha and I investigated the efficacy of anaerobic digester effluent as a use in fertilizer and hydroponics in the summer of 2024 as part of the Barbados Interdisciplinary Tropical Studies program. Here is a short video that sums up our research and our project. Enjoy. Anaerobic digesters use microorganisms to break down biological materials in the absence of oxygen, resulting in methane and a byproduct called digestate. We investigated the use of this nutrient-rich sludge as a fertilizer in both hydroponic and potted settings, using grass-based digestate from the University of the West Indies small-scale digester under Dr. Holder's research project. Cabbages germinated from seed were used as the test species. We chose to use a nutrient film hydroponic system before building and created sketches. Materials used were mostly what was available on site, keeping the budget low. After acquiring most of the needed materials, the grow pipes were cut to 3 feet in length using an angle grinder. 3/4-inch PVC pipes were used as a structural pipe to connect the grow tubes and were capped inside so water would not flow through. 1/16-inch tubing was used to connect the pipes for flow. A hand drill and heat gun were also used to insert the small pipes, tubing, as well as the grow baskets, which were each placed 12 inches apart on center. During the assembly process, plumber's epoxy glue was used to seal the parts and prevent leaks. Post-assembly testing was done to verify the efficacy of the system. The hydroponic runs on a peristaltic pump. Post-construction water was run through the system to test for leaks and to test the flow rate. Bubbles formed as air evacuated the tubes. For this project, we chose to use coconut coir as the growing medium, as it provides good root support and was easily obtainable. We dried the coconuts and harvested the material ourselves. The digestate used in the hydroponic nutrient film, as well as in the potted cabbage experiments, was harvested from the biogas reactor on the UE Cave Hill campus. This digestate particularly came from a feedstock of grass clippings from the island of Barbados. The digestate was harvested by hand and subsequently mixed with water to create the tea that would be used in the hydroponic. It was then filtered to 300 microns before being diffused into the system via the peristaltic pump. The transplanting process of the cabbage seedlings into the hydroponic had a few steps. Firstly, the seedlings were harvested from the original grow cells they were located in. They were then gently washed to remove all of the soil and then gently placed into the coconut coir. The growth of the plants was measured over the course of 6 weeks. Of the 9 samples that were planted into the hydroponic system, only 3 survived the entirety of the duration of measurement. The plant growth was measured by both leaf count and by vegetated surface area. Those that did survive are showing promising growth and have significant root development. Some of the suspected causes of death for those plants that did not make it include lack of oxygenation of the roots, poor light accessibility, pests, as well as potassium deficiency. The potted plant experiment measured 4 different concentrations of digestate: 0, 25, 50, and 100%, with 100 being 1 kilogram of digestate. 4 pots of each concentration were planted and placed in a 4x4 grid with different concentrations running diagonally. This was done to have varying levels of light for each concentration, as the area where the pots were stored had uneven partial shading. These plants were watered daily. It was seen that plants with 0.25 kilograms of digestate grew the best, suggesting that digestate may be a local option for use as fertilizer to augment soil quality. Near the end of the experiment, some pest issues arose, with leaf miners present in 2 of the plants and one of the other plants being completely eaten by what is assumed to be snails. In conclusion, further investigation would be needed to definitively determine if digestate is a suitable fertilizer in Barbados, but this experiment's results show promise. We would like to thank Dr. Lefsrud for his expertise in sensors, the entirety of the BIT staff for their support, and Dr. Holder for his time, guidance, and continuous support with this project.